start, a few words about myself. So I originally studied something called nanostructural engineering. I did my master's in it, but doing that, I get more interested in biophysics or medical-related questions. And in my master's thesis, I worked in optogenetics and electrophysiology, where then I also get my first contact with optical methods, what I will talk about today. And after a quick tour into industry, I came back to university, this time to Leipzig, to do my PhD in a group of Professor K's, which focuses on physics of cancer. Very exciting topic. And we also use a lot of microscopic techniques and even also rheology. And this is where I met Ingolf and his elastography group, and that I get really excited about MRE. And now I'm here and join them. So then about you, if I understand it correctly, you are fresh PhD students, and most of you have a background in physics, right? There is someone with biology, medicine, anyone? Pharmacy. So perfect. So most of you, I think, heard some lectures about optics, I assume. So here, my approach was to give you like a practical insight in very biased topics, rather than go into theory that much. Anyone has experience with tissue or cell culture? Nice. So I'll cover it a little bit later. But since this is kind of focus on life science and medicine, I will start with the most prominent images you will see usually. And this is also something Anna also introduced very nicely. It's H&E stains. We have this twofold staining of hematoxylin, which makes the cell look purplish blue. Hyacin, which makes the ECM, it's the matrix, it's the plastic. And of course, you have different shades of mixed colors from both. And with that, you get this standard H&E images like this. Here you can see an example from cancer. This is metastasis from a colorectal cancer growing in liver. And you can clearly distinguish, even if you are not all pathologists, that here we have different structures than here. So this is the tumor, this is the normal liver. And if you zoom in, you can also see the purplish nuclei and the elongated cytoplasm. And there you have this kind of pushing away boundary. Then you can see it from the image that the tumor pushes away the healthy liver tissue. And in this paper, they also look at different boundaries. So here we have an open, more diffusive boundary. You cannot see the difference between the tumor and the healthy liver anymore. And as you might imagine, this is the much worse case for prognosis. So this is prone to metastasis. But here, as I said, you have to use this H&E stains. If you don't use them, cells look pretty bland in microscopy. This is just a basic bright field image of a cell on a very good imaging conditions on a flat glass surface. And this bright field imaging gives you contrast based on the absorption, which you see or don't see here much details. So if you want to increase that, you can use phase contrast images with imaging, phase contrast microscope, which in contrast exploits the phase shift of the light that goes through the sample. Then you have some special optics like condenser rings and some phase rings in the objectives, and then you get images that look something like this. Better, but also with artifacts. You see this bright hue around the cell, but at least you could distinguish, for example, the nuclei better from the cytosol. But in most cases of more advanced images, in imaging, you have to use fluorescence, which Anja also introduced and explained before. I think you all know how it works. And with that, you have special fluorophores dyes. Here, I also use the example of GFP because it's, as she already told us, it's the most prominent one from this jellyfish. It's a green phosphate protein. So you have an absorption window. This is in the purplish blue, and an emission window, which is in the green. So when you do microscopy, fluorescent microscopy, you also do filter sets accordingly. Because you want to get that light on your sample and that light in your detector. So yeah, but it's all basic microscopy. If you want to go a little bit more advanced, better resolutions, one way to do this is by using confocal system. Has anyone worked with confocal microscopy? Good. Yes. Here, for the other ones, there's the basic principle. So what you have, the problem you have in standard microscopy, that you put a lot of light outside of your focus and also record a lot of light outside of your focal plane. By using confocal microscopy, you kind of avoid it. And there you have, so I think the most prominent compartment is this pinhole device. So basically just a small hole, which blocks out light that is out of the focal plane. So here we have the focal plane, and there we have two light rays out of focus, and you can see they do not go through here because they are focused here or somewhere above. So they pass on to the pinhole. The pinhole has the same focal point as the focal plane you want to image. So they are both in focus, and that's why you call it confocal. And usually you also have a pinhole here in your laser. So here you're not using white light, you're using laser for higher light power and more specialized excitation, I guess. Which also, when you have a pinhole here, also limits the illumination volume. And with that, you can go from fluorescence images, as you can see here, it's just white field, to much more defined laser scanning confocal microscope images. Yes, by the way, I use a lot of information from this Zeiss campus here, which is a very good resource if you want to read up on what kind of microscope. And usually you call this confocal microscopes CLSM. This is confocal laser scanning microscope. Scanning because you just illuminate a very small pixel of your sample, and then you have to move the laser around, usually in a line pattern. And then since you're only imaging in a very small focal region, you then can shift the imaging region up or down and do multiple slices imaging. With that you can get 3D images. And as I said, it's usually dependent on stains, but you can also do reflection imaging with that, which is also cool for certain samples. That means basically instead of having this dichromatic mirror thing here, you just set your detector to the same wavelengths as your laser and record all the reflected light into your detector. Which, you need to have a detector. But I think most of the CLSM have that. And and another prominent mode that captures a similar principle is called spinning disk microscopy. So it's the same principle that I have here, this pinhole device, but now I have it fixed. So basically fixed size and on a rotating disk. With that, I have the possibility to illuminate multiple of the small dots at a sample at once. And this fixed disk is this disk is rotating the defined speed, and with that I get much faster confocal images than with the CLSM. So then it looks something like this. If the disk bit is 0, if you synchronize the disk, then you also get an image of the cell. Big advantage is faster images, but also I must say that modern confocal experiments go out quite fast. And I think you have less light intensity here. The disadvantage is that you have fixed pinhole size, so you cannot tune the pinhole size. That's maybe something I should have mentioned before. So when you go back here to this pinhole, naturally the smaller and make it the more defined my focal volume gets, smaller it gets. And on the other hand, I will also lose a signal. My image, so it's always a trade-off here. Yes, but with that you can do some beautiful images. Here I borrowed one for my colleague Elian Blaut in Leipzig, and she studied how cancer cells accumulate lipid droplets if you co-culture them in samples, fat samples from patients. And here you can see the control, and this is after co-cultivation. You can see this red, this yellow dots here. This is the lipid. In blue, we have the cell nuclei, and in pink, we have the actin cytoskeleton, which was also supporting structure in the cell. And I also wrote down the stains she used here. And you probably noticed that these wavelengths do not match the colors you see. So most of these fluorescence images you see in papers or so are usually colored. Here, two of the stains are in the red, in the let's say normal red and the dark red. Basically, as Anja mentioned before, the longer the wavelength, further we can go in, the energy, the less energy we need, which is less imaging for samples. So the next thing I wanted to talk about is a bit about collagen imaging. We also heard about collagen before. Here I wanted to provide a little context in the context of collagen and cancer, or ECM and cancer. And here I showed you an old representation or simplified representation of the so-called metastatic cascade, meaning when you have a primary tumor and cells can actively migrate outwards of it, they have to go through the extracellular environment to reach blood vessels, and there they can then spread in the body, go out of the blood vessels, and then form metastases. And here they potentially also have to go through the ECM. And ECM, the main structural component of the ECM is collagen. I think most of you have heard it also before. Currently quite popular as a supplement for milk, coffee, or whatever, which doesn't work. Yeah. And but here it's also very important structure of the ECM and a nice tunable system we can use in the lab for experiments. And we work with a mixture of type 1 collagen. So type 1, I think, is the most abundant protein, collagen type in the body. And we use here bovine skin and red tail collagen, mix them together, use a buffer system, and then you can polymerize it at 37 degrees and a fixed pH level. But then you can also tune the properties of the collagen gel. And there you can see a bulk image. It's just a milky looking soft gel. And if you zoom in, so this confocal imaging again, you see this structure. And I was also lucky to work with a confocal microscope that has a high-speed scanner, which allows to make three-dimensional image groups in a very short measurement time. And I could capture basically the live polymerization of a collagen gel. This is what you see here. So after a couple of minutes, the network starts to form, and you can see here the buildup of a network. It looks pretty grainy because it was done in this reflection mode I showed you before. Basically, no stains, only scattered light. If you do not want to image that fast and go more for quality imaging, you of course use some stain that here, for example, Tamra, stains everything, but if your sample only contains collagen, it only stains collagen, right? And with that, you get image that looks like this. So you also have a three-dimensional image cube, and you can see really nicely the collagen fibers here. This is from a publication of a colleague of mine. And of course, if you want to do that in tissue, a few other challenges arise because usually chunks of tissue look like this. This is a thing from a breast tumor we got from pathology. So you have to kind of make it transparent, which also Anja explained in the previous talk. So you have to first fixate it, then wash out the fat, and then do some staining, and at the end this reflective index matching. And this is also something I wanted to talk about really quickly, because if you might know from some optics lecture, the resolution of a microscope also depends on the numerical aperture, which basically gives you information of how much light an objective can capture. And this is dependent on the opening angle of the objective, which is kind of limited in most cases, and the refractive index of your system. And this is something you can also tune. a little bit. So here you see standard objectives. So this is the most basic one, basically just air between your objective and the sample. Then you have dipping objectives, which measure completely underwater, which is, I think, kind of a niche, most often used in electrophysiology, like patch clamp, where you have needles that you poke into cells and measure everything underwater. And the most common one is this immersion imaging, at least for confocal microscopy. And this is also an old representation. Usually we use some kind of like this setup, so like an inverse microscope. It means we look from below. We have a very thin glass slide, and then we have a sample which is some kind of an liquid medium. So let's go back to refractive indexes. So the objective glass has roughly 1.5, air has 1. So there you have a big difference. That means you also have a big loss of light here. So what you can do, you can use immersion liquids, for example, like oil, which also has 1.5. And then this glass cover slip usually are also in a defined thickness and with a defined refractive index. So which is also 1.5-ish. And then in the end, you can just try to match your sample. This is you can do that by replacing the water with some kind of glycerol or oil, basically. So you try to match the refractive indices of all your of the lens, of the immersion, of the glass in the sample and the liquid in the well, that they closer together, so you can reduce loss of signal. Modern objectives look something like this. They have more or less complicated lens structure here, and you also have this adjustment rings where you can adjust for different cover slip thickness, and here immersion media like oil and glycerol or something like this. And with that, and with clear sample, you can make very beautiful images. Here's an example from a spheroid. A spheroid are clusters of cells that are grown in wells, for example, and it can be seen as a model for maybe mini tumors, depending what you want to look at. And here you can see a cross section. This is, I think, 100 microns of a cleared spheroid. And usually, if you would not have cleared it, you would see very dark area here in the center. So tissue clearing makes sense if you want to solve details, not on the surface, but inside. And then you get also some nice 3D rendering of your images. And of course, you can also do that on tissues. Here you can see a very nice example of two chunks of, it's a mixed Mullerian tumor. It's a very aggressive tumor of the uterus that are just placed on top of collagen gels for a couple of days. And afterwards, everything gets fixated, stained, and imaged. And you can see here that they kind of interact with the fibers, they pull on the fibers. And you can see this fiber alignment in between the two pieces. And also the cells spread out here. And you can also see this strange grid pattern here. This, of course, it's basically an stitched image consisting of many small images to do not lose any resolution here. Yes. So, but it's all on dead, on fixated tissue. You can naturally also do it live, so with living cells with tissue, and there it gets a little bit more complicated. The people that have worked with cell culture know that you have to keep cells under certain conditions that they kind of stay alive and grow. So usually it's 37 degrees. If you're human cells, people that work with fish cells, I think they're at 25 or something. And they also need a certain amount of CO2. This is necessary because you have a buffer solution, or you have a nutrition medium in which your cells are growing, and this needs to maintain a stable pH level, and this is regulated usually by CO2 buffer. So if you want to have stable environment for long time imaging, you also have to provide this condition into your microscopes. And this is just an example from Leipzig, a confocal laser scanning microscope, and we have this incubation box here, which can be heated up to 37 degrees. And there you have this CO2 supply for your sample. And with that, you can then also do confocal imaging over a long time. This is what you see here. This is a piece of a breast tumor, again, on top of a collagen gel, and it interacts, it kind of pulls on the fibers. You might see, now it's pulling more. And this goes over the course of 5 days or so. So this stuff can stay alive. Yes. So live imaging works. And then so far I just showed you some nice images, methods. And now I wanted to talk about what can we actually do with this kind of information. Because like analysis based on optical methods. Let's go back to collagen. To this high resolution image cube, what you can do, for example, there is have a look at the architectural features, pore sizes, for example. That's what my colleagues did here in this paper. They had some bubble size algorithm that basically puts bubbles into the open spaces. Then the bubbles grow until they hit collagen fibers, and then they kind of switch the origin a little bit around to let it grow even more. And after certain iterations, they always hit collagen fibers, and this is basically your bubble size or pore size. And with that, you get here, for example, you see 2 different kind of collagen gels. This is only red collagen, and this is bovine skin collagen, and this has much larger bubbles, and it's much more heterogeneous. And you can also get nice numbers on them, and you can see here how the pore size changes if you increase the concentration smaller. So what else can you do? For example, we can have a look at vital tumor tissue. This is from a breast tumor, which I just chopped down with a scalpel in millimeter-sized pieces. So you can see here in the zoomed out image. And you can also do staining and confocal microscopy on this still living samples. And they look something like this. Here we only see the nuclei and the actin structure. We have a cluster of cells, of tumor cells, and there we have some elongated cells that look like they are kind of located in the stroma, in the ECM around it. And then we can compare it, for example, with histopathology images. So we're back to H&E stains. This is from the same tumor. This was done by a very experienced pathologist, and he showed us where the border between normal breast tissue and tumor is. And if you zoom in this tumor region, you can also see similar features out here. So we have this cluster of cells. We have some single cells in the stroma, like here. This is a blood vessel, by the way. We also see one in the background. And the cool advantage is that here we have, I think, more details. And especially we can do it in 3D, and we can also do it live. So this is the actual video related to that image. This is a 3-dimensional image cube you look at. It's a little bit tilted. And this goes on for, so this video covers like 8 hours. And you can see a lot of movement in that cluster here, while the cells here stay static. It's in repeat. But you see here, whittling, here don't. So to give some numbers, you can do cell tracking. I just see how the positions of the cells change over time. And you can see that here in the cluster, you have much more activities than you have in this stroma-like regions. So for example, with this kind of data, you can do cell tracking. You can also have a look at spheroids more. So we are back to this clusters of cells, and you can also analyze the dynamics. Here in the study, which was done by my colleague Steffen, who is now in Barcelona at ITEC, they had a look at 2 different kinds of spheroids. They are from breast cancer. So basically, this is our cancer type. This is the control type. And you can see it with the pure eye that you have a different shape of cells here. This looks much more regular shape, and this is much more chaotic, deformed. But then, of course, you can also do this live measurements, what you see here. And does it work? Perfect. So this is just a live recording, and you don't see that much activity going on in this spheroid. When you go to the cancer, you see cells kind of wiggling around, moving around, and even if you look close enough, you see some cells trading places. So much more activity, much more chaos there. And what he also did was fusion experiments. This is just this is not confocal. It's just wide field imaging. So he lets this two spheroids fuse. While they kind of connect here, you can still see two individual roundish blobs. If you look at the screen, it becomes more clear. But also, if you compare it with the cancer type, all the difference is really, really strong. These behave much more fluid-like, liquid-like, diffuse-like drops. And of course, you can even zoom in more and just have a look at the shapes of the cells. So here it's different color coding because it's for a different paper, but they are the same cell lines. So control and breast cancer. And you can have a look at the cell aspect ratio and the nucleus aspect ratio. And you can clearly see here, for example, the nucleus aspect ratio, that the cancer cell clusters have much more deformed nuclei than the control cells. All based on optical methods. And the cool thing is you can also do that in H&E stains, so from patients. And there we also have access to large databases with tens of thousands of samples. And this is what Pablo Gotthard did from Leipzig in this very exciting paper from 2 years ago. And you can see your punch biopsy from breast tumors, and you have this pinkish or these clusters of tumor cells embedded in the stroma-rich areas. And here's a zoomed-in version, and you can basically analyze the nucleus shape and the cell shape here. It's like a short axis, long axis. This gives you the aspect ratio. So basically the same as you see here, but with the spheroids, that's why I put it here. We also have the information about the dynamics. I showed you before, like the Wickling cells in the tumor spheroids. And here we don't have it. Here we only have the shape information. But we have seen here that these cancer cells are much more active and more deformable or elongated than the control cells. And here we can only have a look at the shapes. So why I'm talking about shapes all the time? This is a very specific reason. When you go into politics of cancer, I think you very early will hear something about unshaming in cancer. This is something I'd like to provide a bit of context here. Anyone heard of it? Tissue fluidization in cancer or something? Okay. It's like a theory to describe a tumor progression as a tissue fluidization. So usually cells behave static in the body. And when you are like in a packed environment, and you need to be mobile to move around. So Joseph Kers has this, let's say this comparison. So imagine if you're in a crowded bar, everyone is standing, it's completely packed, and you want to get a beer. It's very hard to reach the bar and get a beer. But if the people start to move around, start to wiggle, start to dance, whatever, it's much easier to squeeze through and get a drink. And this is basically what also is happening here. So in this unshaming, the cells undergo a collective unshaming transition, meaning we go from a jammed, like a static, solid, like tightly packed, so where there's little room for movement, state towards an unshammed, fluid-like state where they can move around and be active. So this is the concept, and the big question is, here just wanted to show you an example of this. This is from the active spheroid I showed you before, so the cancer one. You can see a cell basically wiggling, squeezing through other cells, and you don't see that in the control spheroid, which is in this case jammed, so inactive. And the big question, of course, is what are the driving factors for this? And there are, I think, many prominent theories. I think the most, it started all with the theory of Max B and Lisa Manning, 2015. They first employed this principle of unchaining, which was known for in glassy physics, and they transported to cancer. That's basically in this model an interplay between the cell-cell adhesion, meaning how good the cells can interact with each other, how good they stick together, and the cortical tension. Cortical tension is how good these cells can maintain their roundish shape. And there's still debate what else can we contribute, let's put it like this. But what's clear in all of this, the consequences that the cells that undergo an unchaining transition are much more deformable. And this higher deformability is something we can exploit again with optical methods. And that's why I showed you all this spheroid and H&E data before. So, and here again, this is from the paper of Pablo Gotay. He created this really nice phase diagram. We have in one axis the standardized mean cell area, which is basically the inverse density of your system. And there you have some kind of shape parameter, sinus, with basically cell and nucleus shape. And depending on the shape and the density, you can derive if it's chained or unchained. So here are some examples from breast cancer he analyzed in the paper. And let me make this maybe a little bit more clear. So with that, you now get from static images information about the activity of your cells by analyzing the shapes of the cells. And as I said before, they've done it for many thousands of samples and derived a marker from it, which then, if you combine it with classical diagnosis routines in breast cancer, can improve overall the diagnosis. It's all in this paper. So really cool stuff. Another point here is that it's also something we've seen before, that cells can also interactively interact with the matrix. So there's also this stress fiber-based motility increase. So basically it means that the cells can stick to, for example, collagen fibers and pull along the collagen fibers to migrate. And this is also something we had a look at. So we go back to this piece of collagen, and there basically you can do analysis of how the pixel in the images shift over time. So particle image velocimetry we did here, and this gives you displacement fields, and you can analyze the displacements and see that, for example, at this point the cluster of cells pull the mouse 72 hours, for example. And then we can also just look at how the shape changes. We plotted that in a paper and compared that to spheroid data, and I think it matched in many cases. It's just an example here. This is only about displacements. It would be nice to have some more mechanics-related data on that, or numbers on that. You can also have a look at the strain. So again, we have an experiment of steroids in collagen. They kind of pull on the fibers. You can see there's an alignment. You can analyze the displacements, as I showed you before, and then you can also calculate the strain rate. It's basically gives you a percentage of deformation. And then you can get strain map, like here, and I colored everything that's above 10% red, because we also did some rheology experiments in collagen. This is not optical, this is shear rheology. You basically have a cone and a plate, and you twist it and measure the mechanical response. And there you can also see an increasing strain. And collagen is a non-linear elastic material. It means that if you have low strains, the stiffness does not change that much. But after a certain threshold, the stiffness goes up very high. We used here the yellow condition, and I just put in a number here at 10%. And everything that's above 10%, you can say that this also getting stiffer. So you can see that this cluster of cells, the spheroids in these tumor pieces, have long-range stiffening effects on the surrounding matrix, at least in this lab system. Yeah, and we also measured it with AFM. Where you can see that you have a stiffer collagen around the spheroid. Okay, this was my collagen spheroid interaction topic here. I want to move on to another method that I think somebody asked if you also can measure single cell properties before. I think left. Yeah, this is the optical stretcher, a device that basically measures single cell deformability. The principle is rather simple. You have a microcapillary and you let cells flow through, then you have some lasers that trap the cells, and then another pair of lasers that kind of stretches the cells. And there you have a microscope objective. So you then need just make a video and can also, again, analyze short axis versus long axis and get the elliptic deformation. Anna also showed some data on the RTDC, which is a much more advanced version of this, I would say. Simply. This was the one where she couldn't show the video. She just showed the data evaluation. And yeah, with the optical stretcher, then a typical measurement looks something like this. We have the ellipticity, and then we switch on the laser, the cells get stretched, gets more elliptic, and if we switch it off, it goes kind of back. Not completely to the base, but gets more roundish. And we can also do some kind of rheological model fitting on that to get some active and passive parameters out of this. But usually just have a look at deformation. Here's a practical example. We have a look at, again, it's breast and cervix tumors in the study. And then you first have to dissociate your tissue to get single cells. It looks something like this. And then you can measure them in optical stretcher. And in this study here, we did this for both cervix and breast, and this is only cervix data. And you can also see here that the tumor cells are much more, significantly more deformable than the control cells. I think it was also your question before on last talk, which is at least, yeah, in a first glance, a little bit counterintuitive because we all know these tumors as with rigid lumps, which I think in all of the cases I can remember they actually are. One exception is the brain. But all of these tumor cell measurements, if you measure them isolated, they show that they are more deformable, softer. You see the same result in breast cancer. It's all in this paper. This is also called rigid tumors contain soft cancer cells. But if you think about all this unchanneling stuff and this spheroid data I showed you before, this higher deformability of single cells makes maybe perfect sense. And then if you imagine that you have like a high interactions with your surrounding matrices, or even as this fibrotic background of most body tumors, this can also explain why they are macroscopically stiffer. Parts of it. But here this is single cells. So, and in the last few slides, I wanted to come back full circle because since we are in the department of experimental radiology, and in the elastographic group, let's talk a little bit about elastography. I assumed that you heard something about the principle of MRE this morning. I think it will come later. So the basic principle of MRE or elastography is that you want to measure mechanics. And in this case, you do it by shaking the tissue, and you use some kind of camera systems to analyze the ear waves you induce in the tissue. And in the clinical MRE, we use some specialized MRI sequences to analyze these waves. You can see a wave image on liver, and from that you can then calculate wavelengths and absorption properties, which gives you then mechanics. Mechanical map. Yeah. But usually in vivo we have a very large voxel size of 2 millimeters. And then we can go to an ex vivo measurements where we have this devices scaled down in a tabletop or in a micro version where we have better resolution, but still far away from cellular resolution. And now, quite recently, we have combined elastography with optics by composing this principle towards a microscope. And here comes OMTE, which is the optical multifrequency time harmonic elastography. So we apply the same principles as in MRE or ultrasound elastography to an optical system. That means we can use very flexible optics. So like inversion microscope, upright, or just a macro lens. Then we need some devices that shake our samples, do that by small piezo actuators. See some here. And most importantly, we need in this case a very good high-speed camera because we measure at high frequencies. So this goes from 800 to 10 kilohertz, so way above AFM frequency range. And we need to capture basically synchronized images to our vibration to analyze the shear wave field. We do that with optical flow algorithm based on harmonic conjunct method. This is much more advanced than the PIF I showed you before. And the really cool thing, we can measure really, really fast. So one measurement with multiple frequency takes less than a second. Here you can see Jakob Jordan, who was basically the brain here behind this method, performing a measurement. Unfortunately, the sound is not working. Would have heard a short beep, and that's it. Multi-frequency sweep in less than a second. He was measuring biofilms. You can see synchronized wide field images, and you can see here some movement. After analysis, you can see wave fields. And this you can then basically analyze from mechanical parameters. Since it uses most of the sample excitation, data acquisition, and data evaluation routines we also use in MRE or ultrasound elastography, it's very comparable with our other methods. You can see dots is OMTE, lines is tabletop MRE, they match really well. You can also see spatially resolved structured phantoms. This is 7 Tesla, this is OMTE, look pretty much the same. This one takes 2.5 hours almost, this less than a second. And we can also do it in a little bit more interesting systems like zebrafish, which some of you might work with. They're quite common in research, both in the embryo and adult stage. They're cheap, they go fast. And can be also mapped in 7 Tesla, as Jakob has shown here. This is just the adult fish, but the embryos are simply too small, you just see them. But luckily you see them in the microscope. And so we could do OMTE. This is embryonic zebrafish, 3 hours and 120 hours after fertilization. Here are some wave fields. And from that we get this shear wave speed maps, which looks basically like optical pictures, but let me make it clear. You're looking at a map of mechanical parameters here. So, which is based on an optical method. It's really, really cool. And you can zoom even more in and see how different features of these fish embryos have different stiffness. So with that, I think we came back to the optic elastography, and I think I will close it here. Thank you for listening. Here's the flip.
I'm just curious, how do you culture the spheroids?
How to make a spheroid? Yeah. There are different methods. I think the oldest one is doing them like in a hanging drop. Put cells in liquid and then put it on a surface and just flip it around. And you have a drop, and then the cells accumulate there. I think much more elegant method is to use specialized wells. So what you need to do is that usually these cells like to stick to surfaces, and you need to make the surface less sticky than the cells. And the cells want to stick together. And we use wells that have like this U-shaped bottom, which have a treatment that are non-adhesive. Basically, all the cells collect bottom and form the solids. Yes.
You guys try mixing a tumor spheroid with a normal spheroid?
You mean,
Would that tell us something more about how a tumor behaves?
you mean
They mix two tumors together, right?
so like
Also, why would they fuse together? Like, what's the biological basis behind a spheroid?
very good question. Yeah. So you're not asking about mixing tumor cells and non-tumor cells in spheroids. You want to have a tumor and a non-tumor spheroid.
Yeah, like,
Yes.
wouldn't that tell you something more about how a tumor attacks a normal?
Yes, my colleague Mario Merkel is currently working on that actually, but I have not seen the results. Your other question, why? I
Yeah,
think,
why did they-
yes, it has a lot to do with, I think, the activity, and there are this cortical rings around the non-tumor spheroids, which basically give some kind of a collective surface tension. When you see the image, it becomes very clear. This one, this. So it's here. You have like a collective boundary structure, which is usually much brighter than the other green you see there. If you remember Anna's talk with the lumen organelles, so you also see these bright rings. These are actin cables that kind of keep the cell together, these spheroids together. So they perform some kind of, you can also say it as an increased surface tension. This is basically what Steffen tried to show, sorry, wrong direction, with these fusion experiments, because you can actually analyze them just like liquids, like the fusion of liquids with different properties or surface tension. You can see that you have a higher surface tension here than you have here. It's one way to describe it.
I have another question. So, like with regards to the mortality, right? Like cancer or like tumor cells have a higher mortality, but like that seems to be guided by like cell adhesion and cortex. Like that's the driving factors, right? Can I make any cell more mortal by wearing these things?
I'm not sure if it works with any cell. So in this jamming,
What's special about a tumor cell? Like, why would a cell that becomes cancerous, like with some genetic mutation, want to move? Cells have no concept of moving, right?
Yes,
They
so-
respond to things.
They are experimental system where you can actually switch that on and off. This goes towards your first part of the question. I'm not sure if it works with any cell types, but there are cells that are not tumorous, that you can behave like tumor cells. This goes more into the direction of epithelial versus mesenchymal cells. This is like more, let's say, different cell types.
Yeah.
And
No, I mean,
tumors-
of course, like for example, like you have these immune cells, right? Which
Yes, yes.
attach to ICAMs and PCAMs, and that's how they migrate to wherever the problem is. But those are specialized cells made for motility.
Yes.
Tumor cells can be, any cell can become tumor cells.
Yes, but they have to undergo certain, let's say, checkpoints or so to become motile. I think also one is that they are, okay, this is like a very simplified version, that they are, like to stick more to the surroundings than to other cells. Because if you have like this very high cell adhesion, it's very hard for them to kind of break out and move. Yeah, but I think it's also much more explained in this paper here on Steffen, where it goes about this fluidization, cancer versus non-cancer. Yes.
In the last technique that you mentioned, do you need to know any mechanical property of or any property of the sample in order to have a quantitative information of the mechanical properties?
No, you basically have the frequency to put in, and then you measure the wavelengths, the absorption, and the light speed. Okay, we are assuming a single density.
Okay.
I think we will have then some MRE-based talks, which use the same principle. Yeah. Yes.
I have just a curiosity about the spheroids. Are they doing it with like multicellular spheroids or just cancer cells? Because like mixing them with fibroblasts or other cells that you can find.
Then it gets really complicated. And this one I showed you, this was just from one single cell type. Just a look at non-cancer and cancer. Colleagues of mine did this, also doing this currently in cervical cancer cells. But yeah, of course, you can also mix cell types and stain them differently.
So complicated.
Yeah, it's very complicated. Here it was about basic stuff, about the shapes, how they change, and how it is connected to motility.